Now imagine your brain as a supercomputer, and your neurons as the wires that connect it to the rest of your body. How do these wires transmit information? Let's find out how nerve impulses work. A nerve impulse is a signal transmitted along a nerve fiber in response to a stimulus such as touch, pain, heat, or cold. It is the movement of action potential along the nerve fiber, which is due to the movement of ions in and out of the cell. It serves to transmit a record of sensation from a receptor or an instruction to act to an effector such as a muscle, a gland, or another nerve cell. A nerve impulse is important for the nervous system because it is the means by which a nerve cell communicates with another cell and coordinates the functions of different organs and systems in the body. Now the main steps involved in the transmission of nerve impulse are resting potential. This is the state of a neuron when it is not stimulated. The inside of the neuron is negative compared to the outside, due to the different concentrations of ions across the membrane. The membrane potential is about -70 mV MV equals millivolt. The minus symbol here means that the inside of the neuron is 70 mV less than the outside. Graded potential. This is a local change in the membrane potential caused by a stimulus such as light, heat, chemicals, or neurotransmitters. The stimulus opens ion channels that allow sodium or potassium ions to move across the membrane, causing depolarization, more positive, or hyperpolarization, more negative of the membrane. The magnitude and duration of the graded potential depend on the strength and duration of the stimulus. Action potential. This is a rapid and large change in the membrane potential that travels along the axon as a nerve impulse. It occurs when the graded potential reaches a threshold level about -55 mV and triggers the opening of voltage-gated sodium channels. Sodium ions rush into the neuron, making the membrane potential positive, about +30 mV. This is called depolarization. Then, voltage-gated potassium channels open and potassium ions rush out of the neuron, making the membrane potential negative again. This is called repolarization. The action potential is an all or none event, meaning that it either occurs fully or not at all. Synaptic transmission. This is the process of transferring the nerve impulse from one neuron to another at a synapse. A synapse is a junction between two neurons or between a neuron and an effector cell, such as a muscle or a gland. Synaptic transmission can be electrical or chemical. In electrical synapses, the neurons are connected by gap junctions that allow direct passage of ions and electric currents from one cell to another. In chemical synapses, the neurons are separated by a synaptic cleft and communicate by releasing neurotransmitters that bind to receptors on the postsynaptic cell. The neurotransmitters can either excite or inhibit the postsynaptic cell depending on the type of receptor and ion channel involved. The polarized state of a neuron at rest is when the inside of the neuron has a negative charge compared to the outside, due to the different concentrations of ions across the membrane potential or the voltage difference between the inside and outside of the neuron is about -70 mV². This means that there are more positive ions such as sodium, Nal+, and chloride Cl- outside the neuron and more negative ions such as potassium K+, and large organic molecules inside the neuron. The membrane is more permeable to K+ potassium cations than to Nal+ sodium cations, so K+ potassium cations can leak out of the neuron through channels, while Nal+ sodium cations mostly kept out by pumps. This creates a separation of charges across the membrane, which is the basis of the polarized state. The polarized state is important for maintaining a stable resting potential and for generating action potentials when the neuron is stimulated. This charge difference is maintained by the sodium-potassium pump and the leakage channels. The charge difference across the membrane of a neuron is maintained by the sodium-potassium pump and the leakage channels. The sodium-potassium pump is an active transport mechanism that uses ATP to move 3 sodium ions out of the cell and 2 potassium ions into the cell. This creates a concentration gradient of sodium and potassium across the membrane, with more sodium outside and more potassium inside. The leakage channels are passive transport mechanisms that allow some ions to diffuse across the membrane down their concentration gradient. There are more potassium leakage channels than sodium leakage channels, so more potassium ions leak out of the cell than sodium ions leak into the cell. This makes the inside of the cell more negative and the outside more positive, creating the charge difference or the membrane potential. The sodium-potassium pump and the leakage channels work together to maintain the charge difference at a stable level, which is essential for generating action potentials when the neuron is stimulated. An action potential can travel along the axon of a neuron and reach the synaptic terminal by the following steps. When a neuron is stimulated by a threshold stimulus, voltage-gated sodium channels open and sodium ions rush into the neuron, causing depolarization of the membrane. The depolarization spreads along the membrane and reaches the adjacent region, where it opens more voltage-gated sodium channels. This causes a chain reaction of depolarization that propagates the action potential along the axon. As the action potential moves forward, the previous region of the membrane repolarizes by the opening of voltage-gated potassium channels and the closing of sodium channels. Potassium ions flow out of the neuron, restoring the negative membrane potential. The action potential travels along the axon until it reaches the synaptic terminal where it causes the opening of voltage-gated calcium channels. Calcium ions enter the terminal and trigger the fusion of synaptic vesicles with the presynaptic membrane. The synaptic vesicles release neurotransmitters into the synaptic cleft, which is the space between the presynaptic and postsynaptic cells. The neurotransmitters bind to receptors on the postsynaptic cell and cause a response such as an excitatory or inhibitory postsynaptic potential. Neurotransmitters can bind to receptors on the postsynaptic neuron and cause either excitatory or inhibitory effects on its membrane potential by the following mechanisms. When neurotransmitters are released from the presynaptic neuron, they diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane. The receptors are coupled to ion channels that open or close in response to neurotransmitter binding. The ion channels can be either ligand-gated or G-protein coupled. If the ion channels allow positive ions, such as sodium or an action potential can travel along the axon of a neuron and reach the synaptic terminal by the following steps. When a neuron is stimulated by a threshold stimulus, voltage-gated sodium channels open and sodium ions rush into the neuron, causing depolarization of the membrane. The depolarization spreads along the membrane and reaches the adjacent region, where it opens more voltage-gated sodium channels. This causes a chain reaction of depolarization that propagates the action potential along the axon. As the action potential moves forward, the previous region of the membrane repolarizes by the opening of voltage-gated potassium channels and the closing of sodium channels. Potassium ions flow out of the neuron, restoring the negative membrane potential. The action potential travels along the axon until it reaches the synaptic terminal where it causes the opening of voltage-gated calcium channels. Calcium ions enter the terminal and trigger the fusion of synaptic vesicles with the presynaptic membrane. The synaptic vesicles release neurotransmitters into the synaptic cleft, which is the space between the presynaptic and postsynaptic cells. The neurotransmitters bind to receptors on the postsynaptic cell and cause a response such as an excitatory or inhibitory postsynaptic potential. Neurotransmitters can bind to receptors on the postsynaptic neuron and cause either excitatory or inhibitory effects on its membrane potential by the following mechanisms. When neurotransmitters are released from the presynaptic neuron, they diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane. The receptors are coupled to ion channels that open or close in response to neurotransmitter binding. The ion channels can be either ligand-gated or G-protein coupled. If the ion channels allow positive ions such as sodium or calcium to enter the postsynaptic neuron, they cause a depolarization of the membrane, making it more likely to fire an action potential. This is called an excitatory postsynaptic potential (EPSP). If the ion channels allow negative ions such as potassium or chloride to enter or leave the postsynaptic neuron, they cause a hyperpolarization of the membrane, making it less likely to fire an action potential. This is called an inhibitory postsynaptic potential, IPSP. The postsynaptic potentials are graded, meaning that their amplitude depends on the amount and duration of neurotransmitter binding. They are also local, meaning that they decay as they spread along the membrane. The postsynaptic potentials can be summed up in time and space, meaning that multiple inputs from different synapses or from the same synapse at different times can add up to produce a larger or smaller change in membrane potential. This process is called synaptic integration. The postsynaptic potentials are terminated by the removal of neurotransmitters from the synaptic cleft, either by enzymatic degradation, reuptake into the presynaptic neuron or glial cells, or diffusion away from the synapse. This allows the receptors and ion channels to return to their resting state. One example of a neurotransmitter that can modulate the transmission of nerve impulse is dopamine. Dopamine is a catecholamine neurotransmitter that is involved in reward motivation, movement, learning, and memory. Dopamine can have different effects on the postsynaptic neuron depending on the type of receptor it binds to. There are 5 types of dopamine receptors, classified into 2 families: D1-like receptors, D1 and D5, and D2-like receptors, D2, D3, and D4. D1-like receptors are coupled to GS proteins, which activate adenylyl cyclase and increase the production of cyclic AMP, CAMP. CAMP activates protein kinase A, PCAF, which phosphorylates various proteins and channels and modulates their activity squared. The activation of D1-like receptors usually causes an excitatory effect on the postsynapse. One example of a neurotransmitter that can modulate the transmission of nerve impulse is dopamine. Dopamine is a catecholamine neurotransmitter that is involved in reward motivation, movement, learning, and memory. Dopamine can have different effects on the postsynaptic neuron depending on the type of receptor it binds to. There are 5 types of dopamine receptors, classified into 2 families: D1-like receptors, D1 and D5, and D2-like receptors, D2, D3, and D4. D1-like receptors are coupled to GS proteins, which activate adenylyl cyclase and increase the production of cyclic AMP, CAMP. CAMP activates protein kinase A, PCAF, which phosphorylates various proteins and channels and modulates their activity squared. The activation of D1-like receptors usually causes an excitatory effect on the postsynaptic neuron by increasing the conductance of sodium and calcium channels. D2-like receptors are coupled to G proteins, which inhibit adenylyl cyclase and decrease the production of CAMP. This reduces the activity of PCAT and its downstream targets. The activation of D2-like receptors usually causes an inhibitory effect on the postsynaptic neuron by decreasing the conductance of sodium and calcium channels and increasing the conductance of potassium channels. Dopamine can also modulate the presynaptic neuron by binding to autoreceptors, which are dopamine receptors located on the same neuron that releases dopamine. Autoreceptors are usually D2-like receptors that inhibit the release of dopamine by reducing calcium influx or enhancing potassium efflux in the presynaptic terminal. This provides a negative feedback mechanism to regulate dopamine levels in the synapse. Dopamine can also modulate other neurotransmitter systems by interacting with their receptors or influencing their synthesis, release, reuptake, or degradation. For example, dopamine can modulate acetylcholine by binding to muscarinic receptors and inhibiting acetylcholine release in some brain regions. Dopamine can also modulate serotonin by affecting its synthesis through tyrosine hydroxylase or its reuptake through serotonin transporters. Drugs or diseases can affect neurotransmitter function and synaptic transmission in various ways, depending on the type of drug or disease, the type of neurotransmitter, and the type of receptor involved. Some of the possible effects are: drugs can mimic the structure of natural neurotransmitters and bind to their receptors, activating or inhibiting them. For example, marijuana and heroin can activate cannabinoid and opioid receptors, respectively, by mimicking the endogenous ligands. Drugs can increase or decrease the synthesis, release, reuptake, or degradation of neurotransmitters, altering their levels and availability in the synapse. For example, cocaine and amphetamine can increase dopamine levels by blocking its reuptake or stimulating its release. Drugs can modulate the sensitivity or expression of receptors, changing their response to neurotransmitters. For example, alcohol can enhance the function of GABA receptors and inhibit the function of glutamate receptors. Diseases can damage or destroy neurons that produce or use neurotransmitters, impairing their function and communication. For example, Parkinson's disease can cause the loss of dopamine-producing neurons in the brain, leading to motor impairments. Diseases can alter the metabolism or regulation of neurotransmitters, affecting their balance and activity. For example, depression can be associated with low levels of serotonin or norepinephrine in the brain. Different neurotransmitter systems interact with each other to regulate complex behaviors and emotions by modulating the activity and function of various brain regions and circuits involved in these processes. Some examples of these interactions are: the dopamine system interacts with the serotonin system to regulate mood, motivation, reward, and cognition. Dopamine can affect serotonin synthesis, release, reuptake, and receptor sensitivity, and vice versa. For instance, dopamine can inhibit serotonin release in some brain areas such as the nucleus accumbens, which is involved in reward processing. Serotonin can modulate dopamine release in other brain areas such as the prefrontal cortex, which is involved in executive functions. The acetylcholine system interacts with the norepinephrine system to regulate attention, arousal, learning, and memory. Acetylcholine can activate norepinephrine receptors and enhance norepinephrine release in some brain regions, such as the hippocampus, which is involved in memory formation. Norepinephrine can modulate acetylcholine release in other brain regions, such as the thalamus, which is involved in sensory processing. The GABA system interacts with the glutamate system to regulate excitability, balance, and plasticity of neuronal networks. GABA is the main inhibitory neurotransmitter in the brain, while glutamate is the main excitatory neurotransmitter. GABA can inhibit glutamate release and receptor activation, and vice versa. For example, GABA can reduce glutamate-mediated exotoxicity, which is a pathological process that causes neuronal death due to excessive stimulation. Glutamate can regulate GABA-mediated synaptic plasticity, which is a physiological process that changes synaptic strength due to activity. Neurons can adapt to changes in neurotransmitter levels or activity by altering their receptor expression or sensitivity in a process called synaptic scaling. Synaptic scaling is a form of homeostatic plasticity that aims to maintain a stable level of neuronal activity and prevent overexcitation or inhibition. Synaptic scaling can occur in two ways. Upscaling: when the neuronal activity is low, the neurons increase the number or sensitivity of their receptors, especially AMPA receptors, to enhance their response to neurotransmitters. This increases the synaptic strength and excitability of the neurons. Downscaling: when the neuronal activity is high, the neurons decrease the number or sensitivity of their receptors, especially AMPA receptors, to reduce their response to neurotransmitters. This decreases the synaptic strength and excitability of the neurons. Synaptic scaling is important for maintaining a balance between synaptic plasticity and stability, and for regulating learning and memory. New synapses can form or old synapses can disappear during learning and memory formation in a process called structural plasticity. Structural plasticity is the ability of neurons to change their shape, size, and connectivity in response to experience or activity. Structural plasticity can occur in two ways. Synaptogenesis: the formation of new synapses between neurons. Synaptogenesis involves the growth of new axons and dendrites, the extension of filopodia and lamelypodia, the recruitment of synaptic vesicles and receptors, and the stabilization of synaptic contacts. Synaptogenesis is influenced by various factors, such as neurotrophins, growth factors, cell adhesion molecules, and synaptic activity. Synaptic pruning: the elimination of existing synapses between neurons. Synaptic pruning involves the retraction of axons and dendrites, the loss of filopodia and lamelypodia, the removal of synaptic vesicles and receptors, and the destabilization of synaptic contacts. Synaptic pruning is influenced by various factors such as apoptosis, autophagy, microglia, complement system, and synaptic activity. Structural plasticity is important for optimizing neuronal circuits, enhancing information processing, and consolidating long-term memories. In conclusion, this video explains how nerve impulses are transmitted from one neuron to another at synapses using chemical messengers called neurotransmitters. The key takeaways are: neurotransmitters are stored in synaptic vesicles and released when an action potential reaches the presynaptic terminal. Neurotransmitters bind to specific receptors on the postsynaptic membrane and cause a change in the membrane potential, either excitatory or inhibitory. Neurotransmitters are removed from the synaptic cleft by enzymatic degradation, reuptake, or diffusion to terminate the signal. Neurotransmitters can have different effects depending on the type of receptor they bind to and the type of ion channel they open or close. Neurotransmitters can also modulate the activity of other neurons or neurotransmitter systems by binding to autoreceptors or heteroreceptors. Thank the audience for watching and invite them to like, share, comment, and subscribe.